Our fourth talk this afternoon, ladies and gentlemen, is by Connie Liu from Cherry Creek High School in Greenwood Village, Colorado. And she'll be speaking on therapeutic use of microRNA 200C to treat ovarian cancer. Let's have Connie up.
Good afternoon, everyone. My name is Connie Liu, and today I'll be presenting on the therapeutic use of microRNA 200C to treat ovarian cancer. The main difference between a microRNA and a regular RNA is that microRNAs are a regulatory class of non-coding RNA molecules. Scientists used to call them junk DNA because they did not follow the usual pattern of DNA goes to RNA goes to proteins. However, even though they do not code for proteins, they can still regulate them by inhibiting the translation of protein from the messenger RNA or by degrading the messenger RNA entirely. Now they're known to be extremely important in development and diseases such as diabetes, cardiac disease, and what I looked at, cancer. MicroRNAs are key regulators in numerous cellular processes such as cell growth, cell death, and cell movement. However, because microRNAs are often lost in cancers, these cells lose a very key regulatory mechanism, and then they begin to divide exponentially, they don't die when they're supposed to, and they begin to move erratically all throughout the body. These cells become out of control, and they become cancer cells. The cancer that I looked at, ovarian cancer, is the 5th most common cause of death for women in the United States, and it has a relatively low 5-year survival rate at advanced stages, which is when the ovarian cancer cells move from the ovaries and fallopian tubes into the tissues such as the abdomen, the intestines, and liver. Current treatments now include surgery and chemotherapy, neither of which are always particularly effective. Surgery, because it is difficult to remove all the cancer cells from those tissues, and chemotherapy, because cells are now becoming resistant to drugs, including the one that is most commonly used to treat ovarian cancer, paclitaxel. The background which led me to my research was the restoration of a protein by a specific microRNA called 200C. So for normal epithelial cells, 200C is very highly expressed, which leads to the repression of this transcription factor called ZEB1 and brings E-cadherin, this protein, back up. As indicated here, E-cadherin is literally the bond which allows these epithelial cells to stack together in a very organized and very nice manner. Cancer cells, on the other hand, have very low to no expression of 200C, which leads to an upregulation of ZEB1, and E-cadherin goes down. And now we have a problem. As indicated here, the cancer cells are already shifting, they're jumbling, and they're moving. And this movement is the first step in metastasis, which is a cancer hallmark. Therefore, I wanted to see if I restore 200C to these Hay cells and restore their levels, would these cells behave less aggressively? And so, although the cells that I worked with, the Hay cells, are a very aggressive type of ovarian cancer whose main characteristics include extremely fast metastasis, they move throughout the body very rapidly, and they're notoriously unresponsive to the drug paclitaxel. And so I transfected my cells with a 200C mimic, called them my 200C cells, and I also had 2 negative controls, my mock and my negative. My negative were just Hay cells that had been transfected with a scrambled microRNA, which should not actually affect the cells. I then wanted to look at 2 main cancerous characteristics, migration, or the ability of these cells to move using a wound healing assay, and these cells' sensitivity to the drug paclitaxel with a clonogenic assay. There were, however, some very interesting and promising results from the clonogenic assay that resulted in the unexpected initiation of my phase 2, my 2nd year, which I'll talk about in a few minutes. So here's my wound healing assay, and I just had a plate of single-layered cells. I dragged a wound through it and recorded the amount of time it would take for these cells to come back in and close that wound. And here are the results. As we can see here, at 23 hours, the mock cells have completely closed that gap. However, for 200C cells, there's still a rather sizable wound, indicating that the 200C cells did indeed move slower. And here is a quantitative analysis of the wound healing assay. And indicated here by this blue line, we can see from the slope that the 200C cells moved at a significantly slower rate than my control cells. Here's my clonogenic assay, where I plated my cells, added various concentrations of the drug paclitaxel, allowed them to grow for 7 days, stained them, and then counted the surviving colonies. Here are my results, and I would like to draw your attention especially to this graph here, where we can see that the 200C cells were significantly more sensitive to the drug paclitaxel, which indicates that the treatment was more effective and that more of these cancer cells were killed as a result. Here is just another measurement of that same assay, and the total colony size, we see that the 200C cells, the sizes were significantly smaller, indicating that more of these cells, again, were killed. Which leads me to my phase 2. Now that I have the results from phase 1, I wanted to see what additional changes were incurred after 200C was restored. And the two that I looked at were adhesion, or the ability of these cells to stick to different substances, and proliferation, the ability of these cells to divide. Another topic that I want to delve more deeply into was the exact mechanism that was responsible for increased chemosensitivity that I observed in phase 1. And for this, I wanted to look at the level of a gene called TUB3 before and after 200C was restored. So here's the adhesion assay that I designed and conducted specifically to answer the question raised in phase 1. And here are the results, where we see that the 200C transfected cells were actually less adherent to all of the substrates that I tested. And for the proliferation, we see again that the 200C cells divided at a significantly slower rate, which indicates that at the end of a 5-day trial, the overall population was also smaller. Which leads me to my clonogenic assay, where I looked at the gene called TUB3, or class 3 beta tubulin. TUB3 is a gene which encodes for an isotope of tubulin that is not normally found in normal epithelial cells, but usually only in neurons. TUB3, however, is very overexpressed in cancers that are resistant to drugs such as paclitaxel that kill these cancer cells by preventing them from dividing. Therefore, I wanted to see if I transfected these cells with extra or exogenous TUB3, but with a non-functional 200C binding site, would these cells still exhibit similar characteristics I observed in phase 1, including increased chemosensitivity? And so for my clonogenic assay, I transfected all my cells with a 200C mimic. I set aside part of them to be my control cells, and then transfected the other group with this extra TUB3. We actually see that the TUB3 cells were significantly less sensitive to this treatment, and so fewer of these cells died when exposed to the same concentration of paclitaxel. And here, again, we see a similar trend, that for the total colony size, the TUB3 cells were indeed much larger. And so for phase 1, I noticed that the 200C cells migrated slower, which is an indication of less aggressiveness and less invasiveness. This is especially important for ovarian cancer cells that metastasize so quickly throughout the body, because a decreased invasiveness could indicate that these cells would concentrate in a smaller region, making the treatment that much easier and decreasing the harm done to patients during that treatment. 200C cells were also much more chemosensitive to the paclitaxel, again, an important benchmark for treatment. So the interesting observations from the adhesion prompted me to conduct my adhesion assay in phase 2, where I noticed that the 200C cells were less adherent to all of the substrates that I tested for. And this, again, is promising because these Hay cells behave less cancerously, they would be less prone to form larger tumors. And so smaller tumors could also prompt a more effective treatment. The 200C cells also consistently proliferated or divided at a slower rate, so there will be fewer cancer cells to kill in the first place. Cells with high TUB3 levels were also more chemo-resistant to paclitaxel. And so remember, normal epithelial cells have high expression of 200C. They repress this gene called TUB3, and the cells die when treated with paclitaxel. Cancer cells, on the other hand, have very little to no expression of 200C, which leads TUB3 comes back on and leads to chemoresistance. So now that we've established this, we should move from cell culture and into animal testing, first in mice, and then into human trials and clinical studies. However, first we need to figure out a way to move from manipulating these cells in the lab, where it is easy to restore this 200C levels, into actually transfecting these microRNAs into already existing cancer cells in cancer patients. And so the 3 delivery methods I researched were lentiviruses, lipid cations, and nanoparticles. The lab in which I'm working with are actually already using lentiviruses to observe the effects of microRNA in mice. However, the last 2 are actually preferable, especially when we go into human trials, because they are non-viral methods. And remember, all of this is with the ultimate goal of dramatically improving the survival rates of cancer patients. So further research into microRNAs could dramatically alter the course of cancer treatment in 3 fundamental ways. First, it could increase the effectiveness of current treatments such as surgery and chemotherapy, and it may even eliminate the necessity of surgery altogether. Secondly, microRNAs could provide an alternative pathway that would completely bypass some of these cells' resistance to drugs such as paclitaxel. And lastly, microRNAs could create an entirely novel and very powerful class of therapeutic strategies that would be extremely influential in interfering with both cancer initiation and progression. And so I hope that in the future, microRNAs could prove to be could play an extremely key role in our fight against not just ovarian cancer, but against all cancers. And so I would like to thank the Rigger Laboratory and the University of Colorado Health Sciences for providing all the facilities necessary for my research, Siemens Foundation and College Board for allowing me this opportunity to present, and George Washington University for hosting this event. Here is a partial list of my references, and thank you very much.